Hello, science fans. Welcome to the Curious Scholarette. Today we're going to talk about something truly amazing. It involves a tiny insect, a deadly disease, and some of the most cutting-edge science on the planet. For centuries, humanity has been locked in a battle with malaria, a disease carried by mosquitoes. We've used nets, sprays, and medicines, but the mosquitoes keep fighting back. It's a huge problem, but what if we could change the mosquitoes themselves? What if we could use their own biology, their own DNA, to stop the spread of this terrible illness? It sounds like science fiction, doesn't it? But it's not. It's a real technology called a gene drive, and it could change everything. Imagine a world where a mosquito bite is just an itchy nuisance, not a potential death sentence. That is the world scientists are working towards. This isn't about creating a new bug spray or a new pill. This is about rewriting the rules of the game at a fundamental level. We are looking at a way to make mosquitoes our allies in public health rather than our enemies. This bold idea represents a massive leap forward in our ability to tackle one of the oldest and most persistent diseases known to humankind. It's a powerful concept, one that brings with it incredible hope but also a great deal of responsibility. The idea is both simple and revolutionary. Instead of trying to kill every single mosquito, which is practically impossible, we could release a small number of special genetically engineered mosquitoes. These mosquitoes would carry a new genetic instruction, a drive, that spreads rapidly through the wild population. This instruction could prevent them from carrying the malaria parasite, or even cause the population of malaria-carrying mosquitoes to shrink dramatically. We are talking about using genetics to do a very specific, targeted job. It's an elegant solution to a very messy problem, and it's happening right now in labs around the world. So let's dive in. Let's consider the science, the possibilities, and the important questions we need to ask. This journey will take us into the world of genetics, ecology, and global health. We will explore how this technology works, what the latest research shows, and why scientists are proceeding with both excitement and extreme care. The future of millions of lives could be at stake. Understanding this science is important for everyone, because the decisions we make about technologies like this will shape our world for generations to come. So, let's get started and explore the science of gene-drive mosquitoes. Let's talk about malaria. It's more than just a fever. It's a life-threatening disease caused by tiny parasites. And these parasites have a chauffeur, the female Anopheles mosquito. When an infected mosquito bites a person, it injects the parasites into their bloodstream. These parasites then travel to the liver, where they multiply, and later invade red blood cells. This invasion is what causes the classic symptoms of malaria: intense fevers, shaking chills, and flu-like illness. For many, especially young children and pregnant women, the disease can be severe, leading to anemia, organ failure, and even death. It's a vicious cycle of sickness that has plagued humanity for thousands of years. The scale of the problem is just staggering. Every year, hundreds of millions of people get sick with malaria, and tragically, hundreds of thousands die. The vast majority of these deaths, over 90%, occur in Africa, and the most vulnerable victims are children under the age of 5. Think about that. A child dies from a mosquito bite almost every minute. This isn't just a health crisis, it's a crisis of development, poverty, and justice. When children are sick, they can't go to school. When adults are sick, they can't work and provide for their families. Malaria traps communities in a cycle of poverty and holds back entire nations. For decades, we've fought back with everything we have. We've used insecticide-treated bed nets to protect people while they sleep. We've sprayed insecticides inside homes to kill mosquitoes. And we have powerful medicines to treat those who get sick. These tools have saved millions of lives, and that's a huge victory for science and public health. We should all be proud of that progress. However, the fight is far from over. The malaria parasite and the mosquitoes that carry it are incredibly resilient. They are experts at survival, and they are constantly evolving to outsmart our defenses. This brings us to a major challenge: resistance. Mosquitoes are developing resistance to the insecticides we rely on. It's evolution in action. The insecticides kill the vulnerable mosquitoes, but the few that have a natural resistance survive and reproduce, passing that resistance on to their offspring. Over time, our best chemical weapons become less and less effective. Similarly, the malaria parasite is developing resistance to our best medicines. This is why we are in a constant race against the clock. We urgently need new innovative tools to stay one step ahead of this deadly disease. We need a game changer. So, how can we change the game? The answer might lie in something called a gene drive. Now, this gets into the really cool science of genetics and heredity. Normally, when two organisms reproduce, a baby gets about half of its genes from one parent and half from the other. This means any specific gene or trait has a 50-50 chance of being passed down to the next generation. It's like flipping a coin. This is standard Mendelian inheritance, the basic rules of genetics that we all learn about in school. It's a reliable system, but it means that a new trait can take a very, very long time to spread through a large population. A gene drive changes that coin flip. It's a genetic engineering tool that breaks the normal rules of inheritance. Instead of a 50-50 chance, a gene with a drive system attached to it ensures it gets passed on to almost all offspring, not just half. Think of it as a genetic cheat code. The gene drive machinery, often built using the CRISPR gene editing tool, copies itself from one chromosome to its partner chromosome in the reproductive cells. This means that nearly 100% of the mosquito's offspring will inherit the gene drive. Then, when those offspring mate, they pass it on to nearly all of their offspring, and so on. This supercharged inheritance is what makes the gene drive so powerful. A trait that would normally be diluted or disappear can instead spread through an entire population with incredible speed, sometimes in just a handful of generations. Scientists can design these gene drives to carry a specific cargo. For malaria, there are two main strategies. One is a population suppression drive, which is designed to make female mosquitoes sterile. Since only females bite and transmit malaria, this would cause the local mosquito population to crash. Another approach is a population replacement drive, which spreads a gene that makes mosquitoes unable to get infected with the malaria parasite in the first place. The potential here is enormous. Instead of a constant, expensive effort to spray chemicals or distribute nets, we could make a one-time release of a small number of these engineered mosquitoes. They would then do the work for us, spreading the anti-malaria trait naturally. It's a self-sustaining and highly targeted approach. The gene drive would only affect the specific mosquito species that carries malaria, leaving other insects, like bees and butterflies, completely untouched. It's a precise biological tool designed to solve a precise biological problem. It's truly a remarkable piece of scientific ingenuity. So, this all sounds great in theory, but does it actually work? This is where careful, step-by-step scientific research comes in. Scientists don't just release these mosquitoes into the wild. They test them in highly secure laboratories and controlled environments to see what happens. A recent and very important study did exactly this. Researchers from a group called Target Malaria built large enclosed cages in a lab in Italy. These cages were designed to simulate a tropical environment, complete with the right temperature, humidity, and even pools of water for mosquitoes to lay their eggs. It was a little mosquito paradise, but with a very important scientific purpose. Inside these cages, the scientists placed a population of normal Anopheles gambiae mosquitoes, the main species responsible for spreading malaria in Africa. Then they introduced a small number of male mosquitoes that had been engineered with a gene drive. This particular gene drive was designed for population suppression. It targeted a gene called double sex, which is crucial for female development. When a female mosquito inherits 2 copies of this modified gene, she is unable to develop properly and cannot bite or lay eggs. The goal was to see if this trait would spread and cause the mosquito population to collapse. The results were dramatic. Just as the theory predicted, the gene drive spread rapidly through the caged population. In each generation, more and more mosquitoes carried the modified gene. The number of eggs being laid started to decline steeply. The researchers watched as the population dwindled, and then within a year it happened. The entire mosquito population in the cages completely crashed. They were eliminated. The experiment was a resounding success, demonstrating for the first time that a gene drive could fully suppress a target population of mosquitoes in a large caged setting. It worked exactly as it was supposed to. This was a landmark moment for the field. It proved that the technology wasn't just a concept. It was a practical tool that could achieve its intended outcome under realistic conditions. Of course, a lab cage is not the same as the complex environment of a village in Africa. But this experiment was a critical proof of concept. It showed that the genetic machinery was stable and effective, and it provided the essential data needed to move forward. The results have given researchers confidence that this approach has real-world potential, paving the way for the next phase of careful, community-guided testing. Success of these experiments is incredibly exciting. The potential to save millions of lives is a powerful motivator, but with great power comes great responsibility. Releasing a gene-drive organism into the wild would be a major step, and it's one that raises some very important ethical and ecological questions. People are right to be cautious. What if the gene drive has unintended consequences on the ecosystem? The Anopheles gambiae mosquito is part of a food web. Bats, birds, and spiders eat them. What would happen if we suddenly removed them? Most ecologists believe other mosquito species would likely fill the gap, but we need to be sure. Another major concern is control. What if the gene drive were to mutate? Could it behave in unexpected ways once it's out in the world? Or what if it were to spread beyond its intended population, crossing over into a different non-target mosquito species? Scientists are taking these risks very seriously. They are actively developing safety measures like reversal drives that could potentially undo the effects of the original drive, or drives that are designed to peter out after a certain number of generations. These safeguards are a critical part of the research, ensuring that we have ways to manage the technology responsibly. The conversation is not just about science, it's also about people. Who gets to decide whether to release these mosquitoes? This technology would be deployed in specific communities, villages, and countries. It is absolutely essential that the people who live in these areas are part of the decision-making process from the very beginning. This means engaging in open, transparent dialogue, explaining the science clearly, listening to concerns, and respecting local values and knowledge. Groups like Target Malaria are working closely with communities in Africa, building trust and ensuring that any future trials have full public support. This isn't just good ethics, it's good science. So what does the future hold? Gene drive technology is one of the most promising new tools we have in the fight against malaria, but it's not a silver bullet. The best approach will likely involve a combination of strategies: gene drives, new vaccines, better medicines, and continued use of bed nets. The path forward must be paved with careful research, rigorous safety testing, and deep public engagement. The potential to end one of humanity's oldest plagues is within our grasp. It's a challenging road, but one filled with immense hope. Do you think we are ready to take this step? It's a question for all of us to consider. Like, subscribe, and share if this episode opened your eyes. Stay curious, stay informed.